So this is going to be your whirlwind introduction into immunology. And I usually like to start off looking at the different types of pathogens, and we're going to cover a very, very small subselection of these over the next few days that the immune response has to deal with. And these range from the extremely small, like viruses, up through the very large, such as parasites and parasitic worms that these happen to be schistosomes, which a member of our class works on, but can go into the large roundworms that are several feet long and an inch or two in diameter. And so the immune system has to have in its arsenal a really diverse array of effector mechanisms to be able to deal with this diversity, and also with the lifestyles that come with them. So viruses have a full range of lifestyles, but all viruses by definition end up usually intracellular at some point, and co-opt the cell's own host machinery to perform their replication. And generally, this occurs relatively quickly. You do have chronic viruses, but their replication cycles are still on a relatively short time frame, versus things like bacteria or protozoan parasites, which can be either very fast or also chronic and hide out and undergo extraordinarily long life cycles. And finally, the incredibly complex replication patterns of the more advanced multicellular parasites. This again is schistosomes, and we're going to be talking in more detail about a in-depth analysis of the malaria replication cycle. And so when faced with a pathogen or with any other organism, the immune system has to answer these two big questions. And I think it's a helpful framing for everything I'm going to tell you that these are the questions being answered. And so first question is, when should I respond? So is this infectious? Is it not self? Is it something that's not part of me, which the immune system has to discern in some way? And if it isn't, is it a threat? And most of what the immune system is going to interact with in terms of foreign organisms are not threats, and they have to be tolerated. And so that decision is incredibly complicated and prone to error. And then once the immune system decides to respond, it has to choose out of its arsenal of effector mechanisms how. And so what's the most effective means of control, and how do you limit damage to the host? Because a lot of what goes wrong in a pathogenic response, and this is probably particularly true, though it's still early days to know for sure, but in the cases of severe COVID, are mistakes in answering this question. So the first question was answered correctly, that a response should be made, but the answer to the second question was incorrect. How should I respond? Okay, so we're going to cover, again, very quickly, some very broad themes that, again, I think are useful framing for how you think of any of the really minutiae detailed aspects of immunology. And so one big theme is that any immune response starts with very generalized inflammation, and then as time progresses, it's followed by specific pathogen-directed responses. So you start with breadth, and then you start narrowing it. And a lot of the danger is in how quickly you narrow towards targeting a specific response, and then how effective the pathogen is at getting out of that specific target. A lot of human immunology is characterized, immunology across all organisms is characterized by soluble mediators, cytokines and chemokines that are shared between cells to let other cells know the state of the system and the potential for danger in different areas of the body. Human immunology in particular makes use of these data because they're very accessible, because they can be measured directly from the blood or from tissue samples, usually with a wide dynamic range and a lot of accuracy. Almost every step of an immune response has some sort of feedback regulation to limit hyperinflammatory responses. So for every yes, there's a no that's turned on simultaneously, and the relative amplitudes of those two switches are going to determine the eventual course of the response. There are multiple modes of recognition and specificity determination, and so this inherent redundancy to the system. The immune system will do the same step multiple times with multiple different pathways. And this is really, I think, evidence of this sort of adaptive evolution where the immune system is pulled from a number of different recognition systems and has implemented a lot of these internal redundancies, which sometimes make it very difficult to determine what the critical part of the response really is. And related to that, it's not centrally organized. So it's not a rationally designed system in the sense that you can tease apart exactly what the decision tree is and that it was formed for a particular purpose. And we often speak teleologically about the immune system, and that's very helpful, I think, as an intellectual framework, but it can kind of cloud your judgment in terms of trying to understand why potentially, particularly in pathogenic situations, why something's gone wrong, to think of it as something going wrong from an intended purpose. And so those last few add up to, it's not always clear what is essential, what is redundant, and what is irrelevant. And this is a huge problem in parsing the literature. You can find data that are correlative, particularly, again, when you're looking at human studies, and we always must remember that correlation is not causation. And in immunology, that's a particularly dangerous fallacy. And so when we start thinking about these pathogen-specific approaches, we have to think about what the relative level of danger is in terms of the host and the host response. And these, again, are going to be very general principles that are useful, I think, when faced with a new pathogen or a new area that you're entering. And so some pathogens must be eliminated for host survival. And these are usually these very small, rapidly growing with limited genomes, sort of, not very sophisticated pathogens. And so their primary goal is just to win a race, grow quickly enough to transmit before the host dies or the pathogen's cleared. They don't have a biosensing apparatus. They don't do a lot of, host manipulation beyond usually some very specific targeted pathways to limit general inflammation. And so the best examples of these are things like viruses like influenza, Ebola, RSV. You could put SARS-CoV-2 here. SARS, we'll talk very briefly about it in the virus section, is somewhat more sophisticated than influenza or RSV, but it's the same principle of it wants to get in and get out before there's really a widespread specific adaptive immune response that can control it. And these obviously are pathogens of great economic and health influence. And so the fact that they aren't particularly sophisticated doesn't mean that we've solved their problem. Some pathogens are acute or chronic depending on the host situation. And so these are generally more complicated, larger genomes or an interesting evolutionary history. There's host genetic and environmental factors that might determine susceptibility and permissiveness for chronic versus acute infection. And so this is where TB, malaria to some extent, and some viruses like HCV are at that level. And so the immune system can eliminate them, or they can persist on at a relatively low level. Generally, there's some sort of reactivation phase that make them contagious again, able to be spread. Certainly that's the case in TB. But they can hide out in the host and coexist longer than these pathogens, which basically, these pathogens up here have no off switch, and these do, some sort of dormancy. And then there's some pathogens that have evolved to be almost exclusively chronic, and their goal is chronicity. And so these are generally very sophisticated genomes, usually very large size, but not always. HIV would be an exception there. Their ability to be chronic is not fully understood in every circumstance. And again, a lot of this is because of their inherent complexity. And in some cases, it might just be that we never evolved a mechanism for their effective clearance, but the pathogens never evolved a virulence that made it necessary for us to balance that out. And as such, they're usually not acutely lethal. So they don't want to kill us because they want to live in us for a very, very long time, because their whole premise for transmission is chronic low-level spread. And a good example here are parasitic worms, which you can have for decades, and HIV, which you can also live with for decades. And so at very high level, the way the immune response works, again, is starting from this very generalized, non-specific approach to progressively more and more specific and pathogen-targeted effector mechanisms. And so the broadest and potentially most effective means of limiting infection is just direct removal and physical blockade. And if that fails, then you'll have a level of inflammation that's relatively nonspecific. There might be some effector cells involvement, some professional immune involvement paired with this physical blockade. And if that fails, then you'll have to get to where you have highly targeted, pathogen-specific adaptive immune responses mediated by B and T cells. And so these stages of the immune response are this first physical prevention of infection, either by the skin, mucus, surfactants, cilial beating in your airways and your nose, followed by innate epithelial inflammation. So the epithelium are the stromal cells that make up the structure of your organs. And at the mucosal surfaces, so the lung, the gut, and the skin, these are structural cells that are doing the job of the organ itself. And they will play a critical role in limiting early infections, and so are really should be considered part of your early immune response. If that fails, then you'll still have this generalized non-pathogen specific response mediated by professional innate cells. These are going to be start focusing potentially a little bit more on the class of pathogen, if not the specific pathogen itself, through innate recognition that we're going to talk about. And then finally, if all of these fail, you get your adaptive immune response and clearance of the pathogen by antibody and T cell killing. And there are models that include elements of all of these. And generally, I would say most models kind of follow this order in terms of as they grow more complex, they'll start adding each of these levels. Many models that we'll talk about today have sort of just a generalized immune compartment that is maybe best represented by this generalized innate inflammation. But there are, of course, models, and we'll talk about some that include T cell and B cell activity as well. So just briefly, a few slides on that first kind of physical barrier concept. The skin is your largest immune organ, is something that we often like to say. And you have these important mucosal surfaces where you make a lot of contact with the outside world that can be exposed to pathogens. And so you've got the skin, you've got the lung and the airways where you're breathing in air, and then you've got the gut where you're eating things that can potentially be pathogenic. And so you have a lot of non-specific mechanisms and somewhat specific mechanisms, antibacterial peptides that are secreted that will try to target and limit pathogenic activity. And really just limit anything from getting where you don't want it to go. And so a lot of these mechanisms are purely mechanical. Okay, so if that fails though, and we're not going to spend a lot of time on this level of protection, then you have to start invoking some level of inflammation and immunity. And it's helpful sometimes to think of the immune system in the context of its cellular components, and most of the cells of the immune system are generated through hematopoiesis. And so you've got the innate side of the immune response that's characterized by macrophages, dendritic cells, and these granulocytes, neutrophils, eosinophils, and basophils. And we're going to talk about all of these in a little bit of detail coming up. And these are sort of juxtaposed against the adaptive immune system, which include the B cells and the T cells. And all of these are derived from hematopoietic stem cells. Developmentally, some of these cells, especially macrophages, are derived even before you have bone marrow or hematopoietic stem cells, but they can be replaced by hematopoiesis. Okay, so the bacteria or virus has gotten in, and the physical barricades didn't stop it. And so now we need to recognize that something's in that we don't want to be in. And this is done via these pathways of innate immune pattern recognition. And this is one of my favorite topics in immunology because it's one of the two big areas of immunology. And as you'll see today, both areas of immunology really arose through true hypothesis-driven research. And the hypothesis that was generated to understand innate immune pattern recognition was was proposed in 1989 by Charlie Janeway as the pattern recognition hypothesis, and he wrote this in a purely hypothetical treatise and journal of I think Proceedings of Cold Spring Harbor. And you don't really see these anymore. This is a sad thing that's happened, I think, in biology, is they don't let people really publish hypotheses anymore outside of reviews, and people don't read them anymore or really talk about them. But he really posited this idea that there had to be some form of pattern recognition to explain some of the data that he had seen, but also as a principle of how immune systems maybe should work. And in parallel, Polly Matzinger, who was working at the time in a similar area, proposed the danger hypothesis, which is in many ways very similar and had some features that were shared with the Janeway hypothesis, and also had some distinct features. And there's still some debate over which hypothesis has sort of more credibility now, but the key thing is that both of these big thinkers were really organizing how experimental work was being done towards trying to understand immune recognition. And what drove Charlie's danger hypothesis was what he used to call the immunologist's dirty little secret. And the immunologist's dirty little secret was that any vaccine that had to be made had to include something called an adjuvant. And adjuvants were basically molecular patterns that were included in vaccines either knowingly or unknowingly to mimic the action of the pathogen itself. And when Charlie wrote his pattern recognition hypothesis, we didn't know how those adjuvants worked on the immune system at all. And we knew what those adjuvants were, and we knew innate stimuli that drove immune responses, but we didn't know molecularly how they worked. And so it was a postdoc in his lab that discovered the first element of the system in humans and mice. Some other folks had discovered them in flies. And it was a set of receptors called the toll-like receptors. And so that's depicted here. There are several toll-like receptors, different numbers depending on what species you are, but about 10 in mice and humans. And these are either surface or intracellular proteins that bind patterns, molecular patterns that occur in pathogens that are less common in the host, and so in humans or in mice in this case. And this was the first pattern recognition system that was defined. And then that was followed by RIG-I-like receptors, which are intracellular receptors of single-stranded RNA or DNA molecules, and NOD-like receptors, which are the most recent and largest family defined, and still the most undefined of all of these families. There can be really variable numbers of these, tens to up to 100, and depending on the species. Some overlap here with plants, actually, in terms of their resistant-like proteins. But so all of these pathways activate distinct but overlapping host response pathways, so signal transduction pathways within the cell that are generally pro-inflammatory. So they turn on transcription factors that will then turn on specific gene sets that are pro-inflammatory. And so one of those transcription factors is NF-kappa B, very well studied. Others are the IRFs, which are very well studied and turn on something called the type 1 interferon response. And so all these pathways converge differentially on these central pathways to turn on inflammation in host cells. And these recognition molecules can be on professional immune cells, but they are also largely on stromal cells. And so this is the front line of defense of your mucosal immune system targeted in the tissue itself. Okay. So if we go with a virus, the characteristic response of a virus in response to both toll-like receptors and particularly RLRs, so these RIG-I-like receptors, is to turn on the IRF pathways, and that turns on the production of the soluble mediator cytokines called interferons. And in particular, the first interferons that are turned on are type 1 interferons, which are called interferon alpha and interferon beta. And these can be secreted from epithelial cells, from just tissue cells, and they're initially an alarm to let other cells nearby know that there's a virus in the area, and that will turn on specific transcriptional programs that will limit viral replication in all cells, not in all calls. And it will do things to help promote recognition of infected cells by the professional immune system that we're going to talk about in detail later. This initial release of pro-inflammatory chemokines and cytokines will also activate more professional immune cells, including these innate cells called macrophages and dendritic cells. And these cells can be direct effectors. They can kill things. So they can eat things in their environment and then destroy them. And so this innate activation, either through direct recognition by these three pathways on these professional cells, or through cytokine secretion from other infected cells in the area, will activate these pathways, including phagocytic pathways, which means that the macrophage can go and eat a pathogen. And then it will upregulate chemical destruction pathways inside the macrophage. And so whatever it eats is encased inside an endosome, a membrane inside the cell. And then the macrophage will dump reactive oxygen and nitrogen intermediates into that phagolysosome, and try to kill everything that's in there. We're going to talk about this in more detail when we get to TB and a little bit with malaria. But this is a very effective means of control for both viruses and bacteria. And when pathways like this are disrupted, they can cause greater susceptibility. The reason this is not always on is because this is very inflammatory. So these reactive oxygen and nitrogen intermediates are potentially going to cause damage to nearby healthy tissue. Okay, so then there's this feed-forward amplification, and that was one of those general themes I was talking about. Is now that a professional immune cell has gotten involved, it's going to secrete even more cytokines and chemokines to alert an even greater proportion of the immune system that there's a problem and that they should come to the local area and deal with the infection. And so this is a panel of the classic sort of early stress response that lets you know that there's an infection. So cytokines IL-1 beta, TNF alpha, IL-6, chemokine CXCL8, and the cytokine IL-12 all have different effects on tissue. So they cause loosening of the tight junctions between epithelial and endothelial cells, and this is to allow migration of professional immune cells to a site of inflammation. This is why you swell when you get a cut, or your lungs fill with fluid when you get very sick, because all of those tight junctions are loosening so that more cells can get into the area to try to clear the pathogen. They'll also do things like raise your temperature that can help suppress viral and bacterial replication. But these are also all the things that we associate with feeling sick. And so a lot of treatments now for very severe acute infections are actually blocking these three specific cytokines here to try to limit the damage that they're causing by being systemically produced. Okay, so a lot of what we're talking about when we talk about infections, and the infections that we're going to talk about, are these infections at the mucosal surface, which are at this interface between the outside and the inside of our bodies. And that interface is blocked by some epithelial layer, and that's depicted here. Each of these is an individual cell, and we want to get immune cells. Sorry, I have this backwards. Yes, so this is the epithelial layer, and then this is somewhere in the tissue here. So this is the lung, this is the gut, this is the skin, and so we've got cells pumping along here in the blood, professional immune cells, and we need to get them to where the pathogen is. And pathogen here, I think, is not depicted, which is stupid, but the local tissue has now started secreting these cytokines and chemokines, and they're going to loosen these tight junctions at the epithelial layer, and they're going to allow cells to attach and roll through and get into the tissue where they can now start attacking the pathogen directly. But this is going to come at the cost of letting fluid leak through and causing localized swelling and disruption of the function of that tissue, whether it's your lung or your gut or your skin. Okay, so that's one element of innate immunity. We're bringing these professional cells that are going to eat pathogens and amplify inflammation. There's a whole nother ancient innate immune pathway, and this is part of what I was talking about, these sort of evolutionary collections of seemingly unrelated pathways that are redundant or overlapping in activity. That doesn't get a lot of study. It was discovered decades ago, but and it is studied, but it's just not as popular as it might have been in the past, and potentially as much as it deserves. And that is this pathway called complement, spelled with an E. And complement is the system of soluble proteins that are always circulating in your blood in an inactive form that can be triggered to activate and increase inflammation, and also directly attack pathogens through the activation of complexes of those proteins. And there are multiple ways to activate the complement pathway. So there's what's called a classical pathway of activating it, a lectin-based pathway, and the alternative pathway. The classical pathway requires an element of the adaptive immune system that we're going to talk about later, but that many of you probably heard of, which is antibodies. So antibodies can directly activate complement if they've bound their antigen. So an antibody that's specific for a particular pathogen will stick to it, and it can activate complement. The lectin pathway, lectins are proteins that bind sugars, and sugars are one of those patterns that is potentially unique to pathogens that you don't find in a host. So sugars really distinguish species in profound ways. And so we make proteins that can bind to sugars that we don't have naturally in our body, and that can activate the complement pathway. And then the alternative pathway is that complement itself has evolved to just bind to specific pathogens. So protein sugar complexes on the surface of particular pathogens, particularly bacteria, can just grab complement out of the blood, and that'll start the complement activation cascade. And when complement's activated, it will recruit inflammatory cells. So it acts almost like a cytokine on its own. It will actually coat the pathogen in a way that makes it easier to be eaten by macrophages and dendritic cells, and the cells that eat things. And it can directly kill the pathogens. And the way it directly kills the pathogens is through the formation of this complex called the membrane attack complex. And so complement, it's one of everybody's least favorite topics in immunology, in part because the nomenclature is terrible. I'm not going to go through the nomenclature because it's really outside the necessity for this course, but it's just a series of proteins labeled C1 through C10. And unfortunately, they were discovered out of order, so they don't activate in order. And there are breakdown products of them. So there's C3A, C3B, C5A, C5B. But they're all circulating all the time alone in your blood, completely inactive. And when they get activated through any of these three pathways, the end result of complement activation is the formation of this highly sophisticated complex, which makes these rings of the C9 protein in the surface of whatever the complement's being fixed to, and it will pop holes in that surface. So each of these is a different membrane attack complex made by multiple copies of the C9 molecule, and it just literally bursts bacteria like a balloon being popped with 1,000 needles at once. And when complement activation happens, this can happen in seconds. And so it's an incredibly fast and efficient process, and potentially incredibly dangerous if it is unregulated, but that is why it has this incredibly complex and long cascade of regulatory stop points. This is sort of also a nice example of convergent evolution. We're going to see a strategy of punching dozens of holes in the membrane of an infected cell or of a pathogen later in adaptive immunity, and they're actually totally separate proteins, even though they look like they have the exact same effect. Okay. And then the last form of innate immunity that we're going to cover is what we call natural killing. And so this is now sort of bridging innate and adaptive immunity. So we're invoking a cell type that's called the natural killer cell, which is a type of innate lymphocyte. So we're moving now from the granulocyte macrophage innate cell lineage to the lymphocyte lineage, which we group with the adaptive immune response, but there's this innate arm of the lymphocyte lineage. And it's best characterized and best understood by NK cells. And NK cells are cells that are always patrolling your body and that are looking for cells that have downregulated a specific molecule called the major histocompatibility complex. And this is a molecule that's on every cell, and it's used by your adaptive immune response to surveil the health of a particular cell and tissue. And we're going to talk about it in detail, but it's because it's used for surveillance by the adaptive immune system, many pathogens, particularly complex pathogens, herpes viruses in particular, try to get cells to downregulate these MHC molecules. And how they work is not important for you to understand right now. It's just important for you to understand that a healthy cell has lots of MHC on its surface, and a cell that's infected with some particularly clever pathogens will try to downregulate it so the adaptive immune response can't see what's happening in that cell. And so the purpose of NK cells is to go around and look for the presence or absence of these MHC molecules. And if it sees a cell without an MHC molecule, it gets activated and it kills that cell. And it kills that cell actually with chemicals that make holes in the membrane very similar to these holes that are made by complement. And so this is also a critical arm of anti-tumor immunity for those of you that are interested in that, and is sought to be exploited a lot actually now in tumor immunity. There are other types of innate lymphoid cells that have recently been discovered. So the NK cells are here in this group 1 cell type, but there are group 2 and group 3 cell types that don't seem to have that killing function, but they have this role of making lots of different cytokines and growth factors that will then affect subsequent innate and adaptive immune responses. These cells were only discovered maybe 8 or 9 years ago. And so that just shows you a little bit of how much immunology is still in flux. Hopefully, I usually give a longer introduction at the beginning of this lecture about why everyone hates immunology. I think we've seen a lot of examples of the troubling nature of immunology in terms of its sort of vast nomenclature of numbers of cytokines and cell types, and our love of subsetting cells. One of my old advisors used to say there's never a cell that an immunologist won't subset, and that's generally true. And the key thing I want to convey as we go through very quickly through this introduction and then through the rest of the course is to not be overwhelmed by the nomenclature. Treat it a little bit like you would learning a foreign language, where you just sort of have to immerse yourself in it. You can look up anything you don't know. You're not expected to have to memorize everything on the front end, and just try to really understand the general patterns of effector mechanisms that are represented by each of these discrete, poorly named molecules. This concept of type 1, type 2, and type 3 is going to keep coming back, and you're going to see it mirrored in all the arms of adaptive immunity as well. And so I'll just mention it here, and we'll just keep coming back to it. The type 1 usually involves cell killing and is targeted for intracellular pathogens like viruses and bacteria. Type 2 usually involves tissue reorganization, which is best for targeting large parasitic worms and other large pathogens. And then type 3 targets intra and extracellular bacteria, and is really almost exclusively for those types of bacteria at mucosal surfaces, so many mucosal infections. And the same molecules, the same effector mechanisms that are used by different arms of the immune system can generally be grouped together in these broad patterns. And it's a heartening thing that after years of sort of subdividing, we've been able to start bringing things back together in immunology under these broad categories.